Hello everyone. We will tackle about difference of transcription and translation. What is transcription? Transcription, the synthesis of RNA from DNA. Genetic information flows from DNA into protein, the substance that gives an organism its forms. This flow of information occurs through the sequential process of transcription, which is the DNA to RNA, and translation, which is RNA to protein. Transcription occurs when there is a need for a particular gene product at a specific time or in a specific tissue. Meaning to say, it is the process by which the information in a strand of DNA is copied into a new molecule of messenger RNA. Transcription occurs in the three steps, which are the initiation, elongation, and termination. Let's go to the first one, which is the initiation. Initiation is the beginning of transcription. It occurs when the enzyme RNA polymerase binds to a region of a gene called the promoter. This signals the DNA to unwind so the enzyme can read the bases in one of the DNA strands. The enzyme is now ready to make a strand of mRNA with the complementary sequences of bases. During initiation, the mRNA, the tRNA, and the first amino acid all come together within the ribosome. The mRNA strand remains continuous, but the true initiation point is the start codon. Remember that the start codon is a set of 3 nucleotides that begins the coded sequence of a gene. Remember also that the start codon specifies the amino acid methionine, so methionine is the name of the amino acid that is brought into the ribosome first. The next step makes up the bulk of translation, which is the elongation. And its elongation is the addition of nucleotides to the mRNA strand. RNA polymerase reads the unwound DNA strand and builds the mRNA molecule using complementary base pairs. There is a brief time during this process when the newly formed RNA is bound to the unwound DNA. During this process, an adenine and the DNA binds to an uracil in the RNA. Elongation is just what it sounds like, a chain of amino acids. Grows longer and longer as more amino acids are added on. This will eventually create the polypeptide. Now that we've begun with the start codon, the mRNA shifts a little through the ribosome so that the next codon is up for grabs. Let's go to the third one, the termination. Termination is the ending of the transcription and occurs when RNA polymerase crosses a stop, which is the termination sequence in the gene. The mRNA strand is complete and it detached from DNA. Meaning, the term termination is the state, act, or process of reaching the end or bringing to an end. It denotes to a biological process where a biological entity is being ended or completed. What is translation? Translation is a process in which ribosome and the cytoplasm or endoplasmic reticulum synthesize proteins after the process of transcription of DNA to RNA in the cell's nucleus. The entire process is called gene expression, meaning it is the process of translating the sequence of a messenger RNA molecule to a sequence of amino acids during protein synthesis. Transcription is the synthesis of RNA from a DNA template, where the code in the DNA is converted into a complementary RNA code, while the translation is the synthesis of a protein from an mRNA template, where the code in the mRNA is converted into an amino acid sequence in proteins. That was the difference between the transcription and the translation. I hope you understand. Thank you.